This is a biology class, so what's the definition of biology? It's the scientific study of life. Bio means life, and ology is the study of. Cells are defined as the basic units of life. And later on in the semester, we're going to spend a lot of time learning all about how cells work and what are the parts of these cells. Every living organism, which is a living individual, consists of one or more cells. Cells use DNA to produce proteins, and these proteins are the building blocks for life. Without these proteins, most life as we know it would not exist. So here's a funny picture. We've got a little cat sitting in a colander or something like that, and then we've got a teapot. And looking at this picture, how do we know the cat is alive but the teapot is not? Well, all life shares 5 unique characteristics. And again, this whole 5 situation is how our textbook author presents it. Different books will present this based upon the way that the author organizes these characteristics in their chapter. And these 5 characteristics of life are organization as number 1. Life is organized. Atoms are the smallest chemical unit of a type of pure substance. We also refer to this as an element. For example, the carbon atom is seen here on your screen. Atoms are organized into molecules. Molecules, therefore, are defined as a group of joined atoms. And an example of a molecule, and we're going to spend a lot of time studying this, is DNA. Molecules are organized into organelles, and organelles are defined as membrane-bounded structures that have specific functions within a given cell. And one example of an organelle is a chloroplast. These chloroplasts are found in plants, and we're going to learn all about the characteristics of cells once we get to our next section. Cells contain these organelles, and the cell, as we saw previously, is the basic unit of life. An example here is a leaf cell. So this cell contains all of these different parts that make it uniquely a leaf cell. Cells are organized into tissues. Therefore, tissues are defined as collections of specialized cells that function in a coordinated fashion. And here we can see that the tissue in question is the epidermis. Think epi means upon and dermis is skin. So this is that outer layer or tissue of our plant leaf. Tissues are organized into organs, and organs therefore are defined as structures consisting of tissues organized to both interact and carry out specific functions. One example of an organ in a plant is a leaf. Now you've probably heard this term organ before, right? Human body has lots of different organs. Our heart, our lungs, our liver, stuff like that. Well, plants have organs as well, because by definition, all these organs are structures consisting of tissues that interact. And then organ systems are all of these organs connected together. So in the example of our plant now, we're looking at the above ground parts of our plant. So just the tree trunk and the leaves of this acacia tree. This would be the above ground organ system. They're connected both physically and chemically in order to allow them to function. And we finally got to one organism. A collection of organ systems are what comprise a single organism or a single living individual. Now we're looking at the entire tree, the above ground parts, but also the below ground parts, right? Because the tree has those roots to hold it in the ground and to help it absorb water from the ground so that it can do photosynthesis. So now we're going larger than an organism. Population is defined as a group of the same species of organism living in the same place at the same time. So now we've got a bunch of acacia trees growing in the African savanna. But it's just one single species of tree. It's not multiple species, just one. If we're looking at multiple species of organisms, we're at the community level. This is all the populations that occupy the same region. For example, it's not just the plants, but it's the animals. All of these different organisms interacting with one another in this African savanna. Ecosystem is a step larger. The ecosystem is both living and non-living components of a given area. So living, that's things like plants and animals. But non-living, that's things like the rocks, the sun, the water. These components of an area that are essential for the functioning of that system, but they themselves are not living. And then finally we get to the big biosphere. This is our global ecosystem, all the interconnected ecosystems of our planet that are connected on both land and water, and this is where life is possible. So throughout these lectures, we're going to see some questions, and these questions are little mini quizzes to get you ready for the exams that we're going to have online. So here's question 1. Which of the following statements is false? You've got 5 choices. Organs consist of tissues. Populations consist of organisms. Molecules consist of cells. Organisms consist of atoms. Or organelles consist of molecules. So which of these 5 choices is false? All right, so pause your lecture if you don't want to see the answer yet. C is the correct answer. Molecules consist of cells is a false statement. We could say that cells consist of molecules, right? But not the other way around. So next, we're going to talk about emergent properties. These emergent properties arise at every level of biological organization, and these are defined as qualities that result from interactions of a system's components. As a case in point, look at this picture on your screen. We can see brain cells, and then we can see those brain cells are interacting, and the emergent property in our human brain is memory. We can remember things around us. But it's not just the brain cells that allow us to remember things. It's these complex interactions and chemical reactions that allow us to remember things. Question 2. Which is the best example of emergent properties? So you've got 5 choices. The first choice is wearing glasses gives you better vision. The second choice is welding metal together makes an office building. The third choice is wearing clothes keeps you warmer. The fourth choice is stacking cups on top of each other makes a plastic pyramid. And the final choice is tying strings together makes a longer string. So pause the lecture now if you don't want to see the answer just yet. B is the answer. The best example of an emergent property from this list is welding metal together makes an office building. By taking all of these metal beams, which by themselves are nothing, we can make this fully functional building with complex moving parts. So now we've checked organization off of our list of the 5 characteristics of life. What's number 2? Energy use. Life requires energy. And to help us understand this, we're going to have to define some more terms. First, we have producers. Producers take energy from the sun, and they can use it to make sugars. And the process by which they do this is by photosynthesis. They can take not only energy from the sun, but also nutrients, such as carbon dioxide from the air. Consumers are defined as those organisms that obtain energy and nutrients by eating other things, either eating plants or producers, or they can eat other consumers. Notice that little squiggly line, heat. Heat is being released as We go from one energy transformation to the next. Decomposers are consumers that obtain nutrients from dead organisms and organic wastes, and these are also an important part of this system. As I mentioned, heat is lost every time this energy gets transferred, and where does this heat go? It goes out into the atmosphere and ultimately out into the universe. All right, we've completed number 2. Number 3 is maintenance of internal constancy. Life maintains internal constancy, and this is really defined by this term, homeostasis. Homeostasis is the process by which a cell or organism maintains equilibrium. A thermostat in your home is a great analogy for how life maintains this constancy. If the room gets too warm, then you turn down the thermostat. That's going to cause a strip to bend, a switch to open, and the heater turns off. If the room gets too cold, then you crank up the thermostat. This internal strip inside your thermostat straightens up, it closes the switch, and the heater turns on. The electricity then goes into that heater to turn it on. Humans have internal thermostats that help maintain temperature homeostasis in their bodies. For example, this woman shivers and adds clothing when she feels cold. Homeostasis is not just about temperature, though. We have to keep balance of all of those chemicals in our body, including things like salt, nutrients, and water. All right, there's number 3. Number 4 in our list of the 5 characteristics of life is reproduction, growth, and development. Life reproduces, grows, and develops. And this reproduction can be defined as either asexual or sexual. So on your screen here, we can see a strawberry plant. This strawberry plant is a type of flowering plant. It produces that classic strawberry fruit that most of you have seen before. And that strawberry fruit is not asexual. When we're talking asexual, we're talking about these green leaves that are growing along stems along the ground. These little green leaves are clones of the parent plant. Sexual reproduction involves fusion of sperm plus egg, and that is how these swans, for example, are able to produce these little baby swans. Another type of asexual reproduction can occur in these fungi. So fungus is its own type of kingdom, and here we can see this fungus produces these spores that allow it to make clones of the parent. And here's another type of plant. This isn't a strawberry, but this is a succulent. It's called crassula, and it's producing little asexual clones of the parent as well. Offspring of asexually reproducing organisms are genetically identical. There is no genetic variation within these clones when compared to the parent. Asexual reproduction is a successful strategy only in unchanging environments. It's not going to allow organisms to adapt to a changing environment very effectively. Another way asexual reproduction is useful is when you need to reproduce super fast. For example, yeast. They're single-celled types of fungi, and they're dividing rapidly here, or budding rapidly, asexually reproducing in their colony. Offspring of sexually reproducing organisms receive genetic material from 2 parents. So they get sperm from dad, and they get the egg from mom. Most plants, including the strawberry we previously mentioned, and animals, such as the deer on the screen, reproduce sexually. Here we can see a coconut that's germinating, and then of course here's the mama deer and the baby deer. Sexual reproduction is successful in changing environments because offspring are genetically unlike either parent. And because they're genetically unlike either parent, they may be able to adapt to that changing environment. It's not guaranteed that they'll be able to adapt, but it's possible. This coconut seedling and the baby deer started out as a single cell. And then they grew, their cells divided, and they developed into multicellular organisms. The strawberry plant, as I mentioned, also reproduces sexually by producing flowers. Flowers are the sex organs of the flowering plants. The flower contains both sperm and egg, and after fertilization can develop into a fruit. All right, here's our third question. Which of the following statements is true about reproduction? The first choice is sexual reproduction creates genetic variation. The second choice is sexual reproduction is most successful in unchanging environments. The third choice is most plants reproduce only asexually. The fourth choice is asexual organisms do not actually reproduce. Or the fifth choice is none of the above are true. And that's always a tricky one. On exams, this can get you sometimes because you have all of these choices and then one of them says, wait, none of these are actually true. So pause the lecture now if you want to consider this before I reveal the answer. A is the only true statement. B, C, and D are all false statements. We've completed our fourth characteristic. What's the fifth? The big E, evolution. Life evolves. Here on your screen, you can see a picture of a snake living in the sand. The scales on this snake look just like these grains of sand. We could say that this organism is perfectly suited to the environment in which they live. How is it that this snake was able to evolve these scales that blend into the sand? Well, the snakes that blend in the best catch the most prey, which is their food. Here we can see it's eating this gecko. Those that get food can survive the longest, and those that survive the longest have the most opportunities to reproduce. We're going to talk much more about this later in the semester, but ultimately, this is natural selection in action. Natural selection is the mechanism of evolution. Natural selection can be defined as enhanced reproductive success of certain individuals from a population based on inherited characteristics. As a case in point, here's a population of bacteria. And these bacteria are single-celled organisms that tend to evolve very quickly. Over time, here we can see one of these bacteria evolved a new trait. And we're going to say that this trait is antibiotic resistance. Antibiotic-resistant bacteria are most successful in the presence of an antibiotic because they have a mutation in their DNA that allows them to survive this antibiotic. We've now completed all 5 characteristics of life, and we need to know these 5 characteristics for the upcoming exam. In review, make certain that you can discuss all the roles of natural selection and mutations in evolution. Next, we're going to talk about the tree of life. The tree of life is how biologists depict evolutionary relationships. When we're creating these tree of life diagrams, it's all about identifying a common ancestor. If we look at this tree of life diagram, it shows platypus and kangaroo and human. And we could say that the common ancestor of kangaroos and humans lived more recently than did the common ancestor of both groups share with platypus. So that is to say, humans and kangaroos are more closely related to each other than either are to the third, which is the platypus in this example. To help us understand how this is possible, we need to learn some more words. And the next term is taxonomy. Taxonomy is the scientific study of both naming and classifying organisms. Species are broadly categorized into three big domains on the tree of life. We have domain bacteria, domain archaea, and domain eukarya. Each domain includes one or more kingdoms, and we're going to look at these kingdoms one by one. This is a current research piece that I found, and just recently, scientists created a tree of life for 2.3 million species, and it's so big that we have to represent it in this circular pattern. This circular family tree of the Earth's organisms is considered the first draft in history of the 3.5 billion year history of how life evolved and diverged on our planet. Now, before we talk any further about the different kingdoms that make up these three big domains, remember the three big domains are the bacteria, the archaea, and the eukarya. We first have to define these terms, prokaryotic versus eukaryotic. I'm going to go back a couple slides for a second. Notice this. We've got this prokaryotes. That includes both bacteria plus archaea. So both of these together form the prokaryotes. Prokaryotic cells are much smaller than eukaryotic cells. Here we can see a small prokaryotic cell that kind of looks like a yellow pill with little hairs on it. And then here we can see just a piece of the eukaryotic cell. The eukaryotic cell is huge compared to the prokaryotic cell. In fact, most eukaryotic cells are 10 to 100 times larger than prokaryotic cells. Another key distinction, prokaryotic cells have DNA because all living things need DNA to make proteins, but the DNA is not organized in a nucleus. There is no nucleus. However, eukaryotic cells, by definition, have their DNA contained within a nucleus. These are some very important characteristics that we need to remember when we're telling prokaryotic from eukaryotic apart. And then finally, a third distinction are organelles. Prokaryotic cells lack organelles. They don't have these membrane-bounded organelles, but eukaryotic cells do. Here's the nucleus, that's one type of organelle. Here's another organelle. Here's another organelle, kind of looks like a hot dog or something. All of these organelles we're going to learn in detail once we get to our cells lecture very soon. Now, before we move on once again to what are the kingdoms of life, I want to talk a little bit about classification. Because we've got all of these tremendous diversity of groups, so how do biologists tell them apart? Well, we use this system here. The classification scheme is as follows. Life is organized into these levels: domain, kingdom, phylum, class, order, family, genus, and species. And as we work our way down this classification scheme, the domain level's very big. There's tens of thousands, hundreds of thousands, or even millions of organisms in a domain. But when we get down to this level, to the genus and species level, now we're looking at very few or even just one single organism. And the system that we use to name every single living organism is called binomial nomenclature. Let's look at the root words here. Binomial, bi means two, nomial means name, and nomenclature is a naming system. Therefore, every living thing gets both a genus and a species name. As a case in point, let's take a look at a plant. So here's a common plant that grows all around campus at College of San Mateo. It's called Mimulus aurantiacus, and it's in the family Scrophulariaceae. Now, first off, you might look at this word and say, whoa, how do I say that word? Well, a little hint, if you say a word with authority, people will believe you and they'll think you're saying it correct. So that was a joke. I know it's hard to tell I'm joking when I'm recording a lecture, but that was a joke. Hopefully you laughed a little bit at home. So notice this family name ends in Aceae. All plant families end in this. So if we see a fancy word with this Aceae on the end, then we know it's a plant family. But we can't just look at this plant and say that's a Scrophulariaceae, because in this family there are hundreds of different species in California alone. The genus is Mimulus. An important note here, genus versus genera. Genus is the singular version and genera is the plural version of this word. So you never use the word genuses. If you're looking at multiple organisms in one genus, we refer to them as genera. However, while it's true, yes, I can say this is a Mimulus plant, that's not enough. Because again, in California alone, there's over 100 different species in the genus Mimulus. Therefore, we have to get to the species level, which is Aurantiacus. And a note about the word species is both singular and plural. So this Aurantiacus is the species name. We never say specie, meaning I dropped the S on the end. We only say species. And of course, for most people, they'll never say a scientific name in their life. Of course, now you're taking a biology class, so you're going to start saying scientific names, but most people would just use the common name. And this one is called sticky monkey flower because the leaves are sticky and the flower looks just like a monkey face, right? All right, so now we can get back into the groups that make up the domains. So first, the prokaryotes is domain bacteria plus domain archaea. And both of these are prokaryotic cells that are all unicellular, meaning they're just made up of one single cell. The eukarya involves lots of different kingdoms, and we're going to need to know these 4 kingdoms on the exam. What unites these 4 kingdoms together is that they are all eukaryotic cells with nuclei. They can be both unicellular or multicellular depending on the group. The first kingdom is called Protista, and this is what we call an artificial kingdom because there's still a ton of scientific work being done about the protists, and in the decades to come, the protists will probably be 20, 50, 100 different kingdoms, depending on the level of complexity. But for now, we can just think of it as one big group called the protists. They can be unicellular or multicellular, and they can be autotrophs or heterotrophs. Let's define these terms. Autotroph. Auto means self, troph means nutrition. So these are organisms that can make their own food. Heterotroph, hetero means other, troph means nutrition. So these are organisms that need to eat their food or absorb their food from the environment. The next kingdom is the animals, and this is a real kingdom that is scientifically classified. All of the animals are multicellular, and all of the animals are heterotrophs by ingestion, so they need to eat their food. Kingdom fungi, these are the fungus, which we probably think of as being mushrooms, but mushrooms are just one part of the fungus life cycle. Most fungus are multicellular, and most fungus are heterotrophs by external digestion. So they have to digest their food outside of their body and then absorb it across their cells. The plants are the next kingdom. They are all multicellular, and most of them are autotrophs, meaning they make their own food. The only exception would be if the plant is parasitic, and then it steals nutrients from a plant that makes its own food. All right, here's our next question. How are the eukaryotes different from the prokaryotes? The first choice is eukaryotes are always multicellular. The second choice is prokaryotic cells never have nuclei. The third choice is only prokaryotes are autotrophs. The fourth choice is only eukaryotes are living organisms. And the final choice is none of the above. None of these distinguish the eukaryotes from the prokaryotes. So pause the lecture now if you don't want to see the answer. B is the best answer. Prokaryotic cells never have nuclei. They do have DNA, but that DNA is just loose, contained within that single cell. When you're studying for the exam, make certain that you know which kingdoms contain the eukaryotic organisms. The last section of this lecture is all about the scientific method. In general, all scientific inquiry follows a standard process. On your screen here, you can see that the scientific method has been broken down into 5 distinct steps. This is a very simplified way to describe the process. In our textbook, it describes it a little bit differently. The scientific method can be described by using this example. Rotavirus is common and causes serious illness in newborn babies. Vaccines might be a cost-effective way to prevent this illness in these babies. Basically, rotavirus causes really bad diarrhea in the baby, and of course, if you're a little tiny human, then diarrhea can actually be fatal because you get so dehydrated. So the first step here is to make an observation, and the observation is maybe vaccines can help prevent diarrhea in these newborn babies. Then we ask a question. Is the rotavirus vaccine effective? Next, we consult prior knowledge. This is the step that we often forget. We don't just move forward blindly. We need to see what other scientists might have written about this. Scientific inquiry has been going on for hundreds of years, and especially in the last 50 years, we've had so many papers published about many different scientific ideas. So after we consult the prior knowledge, then and only then can we formulate a hypothesis. A hypothesis is an explanation for how something works. A hypothesis is not a question. For example, the hypothesis could be the vaccine will stimulate the immune system and will therefore be associated with reduced incidence of rotavirus illness in these babies. Then we make predictions. Predictions are always a if-then statement. If the vaccine is effective, then vaccinated children should have lower incidence of rotavirus illness than children receiving a placebo. Then we can design an experiment to collect and interpret data. For example, researchers need to measure the disease incidence among children in the study, and all those children should be about the same age. Once again, after we do our experiment, consult prior knowledge. You need to see if your results match up with other people's results, which then allows you to finally draw a conclusion. But before you publish your work, the very important step is you must be peer-reviewed. You must have other scientists review your work to make sure that it was scientifically accurate in your reasoning. So while it's nice to see the steps of the scientific method as a nice line of steps or a nice circle of steps. In the real world, it's much more interconnected. Questions lead to observations, observations lead to predictions, predictions lead to new questions, and so on and so forth. The take-home message here is that in science, our findings are always tentative. They can always change when new evidence comes to light. The scientific method can't be just put into a box and handed to another scientist and say, hey, go do science. The scientific method is used differently by different scientists for different questions. And most importantly, science does not prove anything. That's not the way it works. When we read in the newspaper that scientists have proof, or there's clinical proof that X, Y, and Z is happening, that's not what we care about in this class. What we care about, because we're learning how to think like a scientist, is that science allows for the support or the rejection of hypotheses. Science is not about finding the truth. Science is about finding evidence to support our hypotheses. So there's different ways that we can do science, both discovery science or controlled experiments. On the right-hand side of your screen, you can see me doing research as a grad student. I went out and collected flowers all over California, and then I sequenced a little piece of the DNA in the chloroplast of these flowers to try to figure out how they're related to each other on the tree of life. And you can see I'm very excited because I uncovered a very rare species in the desert, and I was doing discovery science. The woman looking at birds is doing discovery science as well, identifying bird species. Now, controlled experiments are slightly different. Well-designed experiments are these controlled experiments, and they include an independent variable, a dependent variable, and a standardized variable. These terms are defined as such, and we're going to do this in the context of the rotavirus experiment with infants. The independent variable is what was manipulated. For example, the dose of the vaccine. The dependent variable is what is measured. For example, the number of rotavirus-infected babies. And 3, the standardized variable is held constant for all subjects in the experiment. For example, the standardized variable in this instance is the age of those newborns. In this table, it shows us the definitions of these terms and examples from our study. But notice this last one we had not yet defined, the control group. The control group is the untreated group. It's the basis for comparison to treatment groups. Control subjects may remain untreated or receive a placebo. Therefore, we can have positive or negative controls. Successful experimental designs include large sample sizes, measurable independent and dependent variables, and a clear control. As a case in point, here is an example. Is a diet high in the artificial sweetener saccharin linked to bladder cancer in male rats? We're going to be reading a lot of graphs over the course of the semester, so let's take a look at these. So saccharin is often found in beverages like Diet Cola, and there is some evidence that saccharin might be linked to bladder cancer. So on this graph, we've got the percent with tumors on this axis, and then parents and offspring on this axis. And notice that there's lots of these saccharin-fed rats with tumors, and then here we've got saccharin-fed offspring with tumors. And this is in males only. Now what about the females? Females, we've got none, and then just a few in the offspring. So when we analyze this data, we need to then test for statistical significance, which is the probability that these results arose purely by chance. So based on this evidence, do I eat saccharin anymore? No, I do not. However, it is important to note that they were fed a diet of almost exclusively saccharin. It would be the equivalent of me drinking 300 cans of Diet Coke in one day. That's a lot of saccharin. Here's our next question. And for this one, we have to interpret a table. What is the dependent variable in the experiment outlined in this table on the screen? Is it A, the virus concentration in the vaccine? Is it B, the number of infants? Or is it C, the incidence of illness, which is measured in number of cases per 100 child years? All right, pause if you don't want to see the answer. C. C is the dependent variable in this question. When we're studying for the exam, make sure that we understand all the components of scientific inquiry and all of the steps of the scientific method. In conclusion, I want to present this case study to help put everything together that we've just discussed in our very first introduction to biology. There was a famous naturalist named Charles Darwin, who is credited as being the father of evolution, as really figuring out in the best way that he could how evolution works. And when he was traveling around the world, he noticed these orchid flowers with these really long nectar tubes. Most flowers don't have really long tubes like this, with a little bit of sweet nectar in the tip of the tube. So he predicted that there must be a moth that has a long tongue that allows it to drink the nectar and in turn pollinate the orchid flower. He based this prediction on his theory of evolution, and we're going to spend quite a bit of time later in the semester learning all components of the theory of evolution. Remember that the mechanism of evolution is the theory of natural selection. 30 years later, the moth was discovered. So even though Darwin never saw the moth, he predicted it existed, and then sure enough, yes, there's a moth with a tongue that allows it to get the nectar in this very unique orchid. Make sure you understand how these observations led Darwin to predict the existence of this long-tongued moth in Madagascar. By thinking like a scientist, you will have success as we uncover all of these biological concepts this semester.